So I better just move it very quickly, indeed. So this is a shift, and this is basic biology. And I'll keep this brief. My lab studies Planarian flatworms, okay? And these are just, of course, always teaching. These are members of the Phylloplatys, which is basically a class of animals that includes all the flatworms, okay? And the one thing you want to take away from this slide is that, first of all, these are very small animals, and everybody likes them. I take them to schools, and even adults will say, they have eyes. They're very cool. But what makes them really amazing is that planarians are amongst the most extreme of organisms that can regenerate. And here's a flatworm that I've cut into 3 fragments. I kept the one in the middle. And in the course of a week, he's grown back his head and his tail. And it begins to behave again, and it can eat. Okay? And in the exterior, they appear very simple. But what's amazing about them, and because it's short, this is a short talk, is that they have many organ systems. But the one we care about the most is that planarians are like a little bag of stem cells. And here's a gene that, suffice to say, everywhere that's purple are cells that are a type of stem cell. Okay? And these stem cells in the animal, which comprise about 15 to 20% of all the cells in the animal, some of them are what we call pluripotent stem cells. Now let me step back for a second for another little lesson, just in case you forgot. Stem cells are really popular because these are cells that can self-renew, so they can clone themselves over and over again. And the other basic property stem cell has is that it can differentiate and make some cell that will do something useful, like a muscle cell. Okay? And what's different about them, and we have many different kinds of stem cells. That's why we can grow the skin back. Okay? We can regenerate muscle, less and less so as you get older, but it's true that we can. What's really important is that they have different levels of potency. And humans, as far as we know, do not have this pluripotent stem cell that can make every type of stem cell in the body. Okay, so planarians, for many reasons, have maintained the cells. I'm going to skip this. But just to remind you that this is for the last 20 years or so, the slide I skipped was the person that actually developed, for the first time, culture human stem cells. Okay, these are becoming, it is almost 20 years already, very important in regenerative medicine because we can use them to model animal development, very important for potentially cell transplantation studies and for drug screening. And this gives us the basis, for example, for trying to treat diseases such as neurodegenerative disorders or try to discover some of the mechanisms that cause cancer. Okay? But this is all happening in a dish. And that's still very important. What planarians can bring into the picture is that we can use them to understand how you can regulate a stem cell in vivo. Okay? To put it in the context then of what we do in my lab, is that this is one of the projects we work on. We're very interested in understanding how a planarian can regenerate the nervous system. So I told you in a week they make the head again. It turns out that they make dozens of different types of neurons, including motor neurons. The types of neurons are involved in disorders like Parkinson's disease. I can go on and on. And they made those in a week. So here's their brain up here, which is 2 lobes, 2 ventral nerve cores, and they maintain and regenerate those. So how do we try to solve this problem in my lab? So another small lesson. We try to make, we basically do lots of screenings, okay? And this is an example of what we call a typical genome-wide screen. So we go through the labor of identifying genes we're interested in, and then we go about to disrupt them somehow, okay? And we find out if they give a phenotype. And I'll tell you what I mean by that, okay? And here's how we do it, because I have to show you cool pictures. In planarians, we can do this very simply by using a technique called Rna Interference, which is a way to disrupt gene function. We feed them this RNAi food mixed with foot liver. I always tell people it's organic calf liver from San Francisco, only the best for our planarians. And we mix it with the food dye, and we trick them into eating this, and we cut them. Okay, we love cutting them and cut them and cut them. Okay, and here's one example of one of those genes that we're interested in. It's the transcription factor, so it's a gene that controls the expression of other genes. The protein does. It's expressed in neurons, and when we cut the planarian, just look right here. Here's a control animal. It makes a nice brain, and if we knock down this gene using RNAi, they make an abnormal brain with mixed-pattern eyes, for example. Okay, so we know this gene is important for the regeneration of the nervous system. And then I got to this good. Here's a project we're interested in. Here's a gene that when we knock it down, the planarians cannot sense light. They lose the ability to detect or process sensory information. And when we expose them to light, they cannot control their motility. Okay? And in this gene, in fact, which is called, it's another transcription factor called SOXB1B, has been implicated in human epileptic disorders. So we have now actually found that this gene is important for the planarians to make their sensory neurons, which they need to detect light, perhaps mechanosensation, etc. And so now to summarize, in this very, actually quite simple picture, the work in our lab has helped in the community to identify genes that take the Planarian stem cells from the pluripotent state to make neurons, for example. And now we're about the business, now that we've done many screens, and I'm really tired of screening. My students are doing it now, okay? Now we'd like to understand how these genes work and contribute to our understanding how these genes might be important for the stem cells to be regulated into making different cell types and giving us the right functions. And so I'll stop there because I couldn't be standing up here, and I don't know about stars, because the stars are the students, the fantastic students and postdocs and technicians that we have at San Diego State and the graduate students. And without their hard work, I wouldn't have any data to show you. And I'd like to thank the California Institute for Regenerative Medicine, which funded some of the work I showed you, and right now the National Science Foundation. Thank you. Questions about planarians? Yes?
So it's a bit of a nutty question.
Wait.
Stanley would get mad if you don't
Yeah, it's
use
a
the
bit of
microphone.
a nutty question, but why not? Can you train planarians to do things? Follow mazes?
No, it's
and that
not.
sort of
In
thing?
fact, it's not nutty at all. I'll just give you a quick history.
That's the first part.
The first part.
The second part
part
is nutty.
Okay, so the answer is, for a long time we've known they can be trained to do simple behaviors, okay? They can habituate, and in fact, 30 years ago or longer, the neuroscience community got really upset about this. But now it's been demonstrated again that you can actually train them and use them to understand mechanisms underlying those simple learning paradigms. So it's possible. Yes.
So then the question is, now you cut them.
Uh-huh.
Do the different parts remember what they learned?
So that was a controversial part of the question. So that's, and it turns out, and I'm not working on this, partly because when I was looking for a job, I decided it was probably astute not to go into that, okay? But it turns out that when McConnell, Richard McConnell, I think it is, I don't forget the first name, did those experiments, he had shown at the time, he had this theory of RNA transfer for memory acquisition. And turns out that those experiments could be repeated, but everybody thought they were, they didn't understand them. But now, to go, without going into details, now that we know about RNAs, such as microRNAs that get secreted, that are involved in actually regulating gene function, it turns out that perhaps there was a mechanism that was unknown at the time that could be used to train the different parts. So it's possible to take the regenerative part and still retain the ability to do those behaviors a little bit easier, perhaps not as well, but again, that's as much as I know about it. So there's some possible molecular explanation for what was observed before.
It seems like the pieces do have some kind of memory of what they learned.
Apparently, yeah, apparently.
Jim McConnell, by the way.
Oh, thank you for correcting me. Yeah. Anybody? Yeah. Ricardo, Richard, just sounded good.
Do the planarians have a lifetime, an age where you can't regenerate them after you cut them because of telomere shortening? I mean, is there a certain age where once you cut them, that's it, and they won't regenerate?
Planarians, when you amputate them, they reset the telomere lengths. They maintain them. And as far as we know, I didn't go into all the details. There's hundreds of different Planarian species with different abilities to regenerate. The ones that we use, which I totally skipped the name, Xochitl mediterranea, they come from Spain. These are asexual Planarians that reproduce by splitting themselves apart. So you kind of open the incubator in the morning and they're having sex by cutting themselves, okay? And what happens is that they do this over and over. As far as we can tell, they're negligibly senescent, okay? I've had the same Planarians for 15 years. Some of them, I haven't tracked the same one. But there's some that do seem to undergo some senescence, and I just always want to be careful because there's something, in nature, I don't think they quite make it forever, right? But at least mechanistically, if you study what happens to the telomeres, they reset the length when they regenerate. So presumably, as they're turning over cells all the time, the cells are constantly young. Lucky them.
Ricardo, could you comment on when you identify genes that play some role that has a particular function in human ailments, right?
Yeah.
Heart disease or whatever. How often is it pretty close to something that you could identify to that function in your small animal?
Do you mean it's that the function will be the same, you think? Or?
So let's say gene involved in epilepsy could influence neuronal functioning or electrical.
Yeah. So we're obviously trying to explore the precise answer to that question. But we do think that these genes, the proteins that they make, their functions are quite conserved. And we hypothesize that these genes probably regulate similar types of genes. So in the case of this last gene I showed, the data we have so far, which is pretty exciting, clearly indicates that it's potentially regulating the same types of, let's say, ion channels or receptors that you find are regulated in human cells. But this is something we're hoping to address. But my take on it is that we'll stumble across things that will have a function that's conserved throughout evolution and relevant to human biology. Yeah.
Thank you.